Well, thank you very much for the invitation and the opportunity to speak here. So today I will talk about the development of our software platform called MobiDic, which stands for Ion Mobility DIA Analysis Toolkit, where we use ion mobility to improve MS sensitivity and duty cycle in DIA SWATH type of data. So one of the key challenges that we are all faced with when we do proteomics using LC-MS is that we are dealing with very complex proteomic samples. And this means we are dealing either with whole cell lysates, where we have 100,000 or more peptide species that we would like to separate and quantify. We are dealing with samples of very high orders of magnitude in dynamic range, or with extremely heterogeneous tissues. So we can already measure relatively complex samples. However, we are often faced with challenges in the area of reproducibility. So can we measure the same analyte reproducibly across dozens or potentially hundreds or thousands of individual samples? This is extremely relevant in a clinical context. We are challenged with the limited sensitivity due to the fact that we don't have an amplification procedure for proteins, and of course, to some degree, with limited selectivity. So when we want to move to larger cohorts or multi-center studies, this kind of reproducibility becomes a real challenge in LC-MS. The sensitivity, of course, goes into the direction of the previous speaker, what Matthias outlined. If we have very little sample amount, if we have sorted cells or even single cells, or also if we want to improve the throughput from currently maybe tens of samples a day to 100 or more samples a day. And finally, the selectivity, of course, relates to the fact that we often deal with isobaric compounds that we cannot separate by mass to charge alone. So having additional dimensions of separation can help separate those compounds. So why are we running into these problems with reproducibility? So this has been one of the key interests in my lab, and one of the key motivations to develop data-independent acquisition or SWATH-MS. And the way we look at the data is really that we look at this two-dimensional plane here spanned by mass to charge and by retention time, where each of these individual dots is an individual peptide precursor. And one of the challenges we have in mass spectrometry is that we would like to sample this space as comprehensively as possible. However, our mass spectrometry instruments are not fast enough to sample the whole space. So the solution to that is something called data-dependent acquisition or shotgun proteomics, where the instrument selects, based on the data that it sees, positions for sequencing. And this is an extremely efficient way of going very deep into a proteome. And with this approach, we are only sequencing in areas where there is actually signal. However, there is two main problems here. One problem is that there are a lot of peptides that are not sequenced and not sampled. And the other problem is that this semi-stochastic procedure is not reproducible across very large cohorts, because every single run will sample at a slightly different position and will sample slightly different peptides. So DIA or SWATH-MS addresses this problem by basically taking this two-dimensional space and dividing it up into little pixels. So here are 4 pixels shown in the mass to charge dimension, and what we do is simply have a fixed precursor isolation window that corresponds to the width of the pixel, and then we sample 1 pixel at a time. Once we reach the top pixel, we start at the bottom again. And with this way, we now solve both of the problems. Firstly, there is no more area that is not sampled, because every single peptide will be sampled with this procedure multiple times and will produce fragment ions in multiple scans. And secondly, there is no more problem with reproducibility because we apply the exact same deterministic sampling procedure to every single one of our runs. No matter whether you do 2 runs or you do 1,000 runs, you will always get the exact same procedure of sequencing, and you will get the exact same MS data. So by using SWATH-MS or DIA, we now have a discretized acquisition scheme that allows us to obtain deterministic and reproducible data acquisition. And it also allows us to perform a complete recording of all fragment ions of the ionized precursors, which means we are not missing any data anymore. And this really leads us to believe that this is the key to solving this problem of reproducibility in shotgun mass spectrometry. However, there is of course one big challenge with this approach, namely that now our LC-MS-MS spectra are much more complex because we are actually multiplexing the fragmentation patterns of multiple precursors in a single MS-MS fragment spectrum. So the way we approach this is by not actively trying to deconvolute these individual spectra, but rather we view the acquired data as a complete digital representation of all the precursor and all the fragment ions that the mass spectrometer with the current technology can record. And then we use this as a big database and we formulate queries into the database. And in our case, the queries correspond to questions of quantitative abundance of individual peptides. So here, for example, if we are looking for a specific peptide with 3 different fragment ions, red, blue, and green, our query would consist of the mass to charge ratio of these fragment ions, the retention time of the peptide, and the precursor mass to charge of that peptide. And we would then go into this complex multiplexed MS-MS spectra, and we would specifically extract the fragment MC traces for these 3 fragment ions and then plot this as a chromatographic trace and interpret the data using a targeted proteomics paradigm. So we would then look for coelution of 6 or more fragment ions and would use this to identify and quantify the peak here. Now, this has been the story so far, and this has been working very well, and we were pretty confident that we can achieve very high reproducibility with this approach. However, there are still several issues with the current approach. And one of the big issues that we usually don't like to talk about so much is that doing proteomics in this way is fundamentally different to the way proteomics has been done, where individual fragment ion spectra were always directly linkable to a precursor. And with our approach, we lose this direct association between precursor and fragment ions because we multiplex the fragmentation. Now, the second issue that is not uniquely a problem of DIA, but generally a problem of the way we do mass spectrometry, is that we are still somewhat plagued by low sensitivity. So if you have 32 swath windows, you only measure 1 swath window at a time. So that means you only measure 3% of the ion beam, whereas 97% of the ion beam does not get measured. And secondly, we are suffering from the intrascan dynamic range. So within a single swath window, we can only measure quantitative differences on an order of maybe 3 to 4 orders of magnitude. So if there are co-fragmented peptides of very high intensity here present when we want to measure our peptide of interest, we would actually not be able to measure that because they get drowned out. So this is where our collaboration with the Matthias Mann lab and with Rudi Ebersold at ETH Zurich came in, where we tried to address these key limitations of the current SWATH-MS approach. And we thought that combining DIA with ion mobility would allow us to address both of these current issues. Now, when we looked at TOF acquisition, we actually did something very similar to what Matthias described in his talk. So we looked at the time scale of our different separation technologies. And one of the things that's very obvious is that the LC separation happens on a time scale of maybe 5 to 10 or maybe 20 seconds. And this means that we need to generate spectra at a faster rate than that in order to capture a peak at multiple positions during the elution profile. So usually you would want to have a cycle time of about 3 seconds to capture eluting fragment ions between 7 and 10 times. However, what we of course realized is that the analysis speed of a TOF is much, much faster than the required 100 milliseconds, right? The analysis speed of a TOF is on the order of 0.1 milliseconds. So what the TOF instrument is actually doing is it's acquiring 1,000 individual scans and then simply averaging all of these scans. And so what we thought we could do is actually leverage this approach and try to use the time during which the TOF is doing its 1,000 acquisitions in a much better way. And this is where ion mobility comes in, because the time scale of ion mobility separation fits extremely neatly between the time scale of LC separation. It's still fast enough to allow us to capture 7 to 10 time points across a chromatographic peak, but it's much slower than the analysis speed of the TOF. And so if we perform the ion mobility separation on a timescale of 100 milliseconds, we can actually get a separation over these 1,000 individual TOF pushes. And now each individual TOF push has meaning because it's associated with an ion mobility instead of simply the same measurement over and over again that we then average. So how do we put this together with data-independent acquisition or SWATH-MS? So our traditional SWATH-MS setup, here shown with 32 windows, uses this pixel-based approach where we measure 32 pixels until we reach the top SWATH window, and then we start from the bottom again. And of course, if you take all of the fragment ion spectra from a single SWATH window and you put them in order, you see these elution patterns of the fragment ions here indicated with the red, blue, and green fragment ion. Now, what happens if we add ion mobility to the picture? Something quite extraordinary. So each individual MS2 spectrum is now becoming two-dimensional. So if we look at this individual MS2 spectrum here and we acquire it with the TIMS device, what happens is that we still have the mass to charge dimension, but now we have an additional dimension of separation, which is the ion mobility separation. And this is the precursor ion mobility. And what you will notice now is that all the fragment ions from our peptide of interest here in color are lined up at the precursor ion mobility elution time. And these interfering black peptides that also produce interfering black fragment ions are now separated from the peptide of interest. So this addresses the first issue of linking precursor and fragment ions again in highly multiplexed MS-MS spectra. So that's the first thing we noticed here. The second thing we noticed is that if you look at all of the precursors that are derived from a single SWATH window, let's say, for example, the 400 to 425 mass to charge window, these precursors do not span the full ion mobility range, which also Matthias has shown in his slides. So this actually means we are not utilizing the complete 100 milliseconds, but actually all of these precursors are bunched up in something between 30 or between 20 and 30 milliseconds. And this allows us now to multiplex DIA or SWATH windows within a single ion mobility separation. And currently for complex proteomes, we perform 4 individual multiplexing steps. So we can squeeze 4 DIA windows in a single 100 millisecond separation, but each of these windows still contains the amount of ions accumulated during the 100 milliseconds. So this gives us a 4-fold increase in sensitivity and speed, and a 4-fold increase in the number of ions we actually get into the detector. So what do we gain by adding ion mobility to SWATH-MS? First of all, we gain this precursor fragment associations back. So our multiplexed, highly complex spectra now suddenly look much, much simpler because we now see separation of the fragment ions by the ion mobility of their precursor. Secondly, we gain increased duty cycle due to the multiplexing from diaPASEF. And finally, we gain increased sensitivity and dynamic range, which relates back to the talk by Chris, where we are now seeing that squeezing 100 milliseconds of ions into an ion mobility elution peak of 2 or 3 seconds actually boosts the signal in each individual TOF push. And this is something we see very strongly, that we get additional sensitivity simply due to the concentration of the ions into a very short amount of time. Of course, one of the challenges we had to deal with is that the data has now gained an additional dimension on top of the dimensions we already had in our traditional SWATH-MS data. So how are we analyzing this diaPASEF data? Here, I would like to present the MobiDic workflow, which consists of 2 parts. The first part is called diaPASEF. It's a Python package that allows data conversion from raw files to MCML, library generation and library alignment in ion mobility and retention time, and data visualization. Secondly, we are using OpenSWATH as part of our pipeline that is consuming these libraries and raw files that are generated by diaPASEF, and we perform extractions and scoring in a very similar manner as we have done with traditional SWATH data. So the first thing we did was we went back to HeLa data. We took a HeLa cell lysate, we fractionated it into 24 off-shell fractions, and generated a spectral library with about 130,000 peptide precursors, mapping to almost 9,000 unique human genes. And as traditional, a spectral library contains retention time and fragment ion intensities, but now, for this particular case, we also have collisional cross-section for all of these 130,000 peptides. And in our simulation software, we can now overlay these individual precursor isolation windows with the position of the peptides in the library. And what we see here is the scheme that we are currently using with 32 windows multiplexed 4 times. And what we see here is basically how we slice up this 2-dimensional cloud of ions into our individual DIA windows. So a single PASEF frame will start up here with a given mass to charge isolation window, and after about a quarter of the time, the quadrupole will switch from a high mass position to a medium mass position, acquire for another about 25 milliseconds, switch again to another medium mass position, and then finally switch to a low mass to charge position. And as you can see, the only ions that we are losing are those out here that elute after switching the quadrupole, or those ions up here that elute before we switch the quadrupole to the new position. And of course, what we did, we looked at the number of ions that we lose, and we could show that using the theoretical simulations of the 2+ ions, we were able to potentially capture 97% of the ions in the library and 88% of the 3+ ions, giving us an overall theoretical maximum coverage of about 92%. So again, this is the theoretical coverage. This only indicates that with our scheme, we will be losing about 8% of the ions that are outside the accessible range of our multiplexing scheme. One other thing we looked at is the signal to noise. And here is one example where we ran the exact same experiment on a HeLa cell lysate without the TIMS device turned on of a particular peptide with 6 transitions. And what happens when we turn the TIMS device on is a very significant cleanup of the peptide signal and a very clear increase in signal to noise. So of course, this is a single example, but then we asked, how does this now look on a global scale? And so what our software is now able to do is doing this type of analysis automatically for the full library of hundreds of thousands of peptides. And the data is now arranged in a three-dimensional space of ion mobility, retention time, and mass to charge. And the regions of interest are three-dimensional pixels, or so-called voxels. So these have a width in mass to charge that is on the order of a few ppm. They have a width in ion mobility, which is in the order of a few % of the full ion mobility separation. And they have a width in retention time, which is about 3 to 5% of the full gradient. And we can now take different views onto the data, which is what we use for scoring in OpenSWATH. The first view is a very traditional view for anybody who has done targeted proteomics, which is a simple extraction of a chromatogram. So we can plot the data as a function of retention time, and of course what we see is a coelution of our fragment ions at a given retention time point. However, additionally, we can now look in the ion mobility dimension, and here we see something very, very similar. So we see a coelution of the fragment ions, the 6 fragment ion traces, as a function of ion mobility at the position of the precursor ion mobility. And so we actually realize that this is the elution time point of the peptide because all of the fragment ions show a co-eluting signal here, whereas these other signals are signals that we are now able to exclude from our analysis. And the last way to look at the data is looking at full fragment ion spectrum, because we have high resolution MS-MS spectra, so we can look at ppm accuracy, we can look at the isotopic envelopes of the data and use this for additional scoring. And currently we use all of these 3 dimensions in our scoring model and then use a machine learning approach to integrate these individual scores into a final discriminant score on which we also perform false discovery rate estimates. So when we do that on our HeLa cell lysate, here acquired on a timsTOF Pro in Martinsried, we used a 200 nanogram HeLa cell lysate injection on a 120-minute gradient. One of the things we realized is the drastic increase of the individual components that I've presented today. So when we started out with our initial experiment where we basically run the experiment without any ion mobility separation, we got a decent number of peptides identified, but nothing to write home about. However, as soon as we turned on the TIMS device, but then deleted the ion mobility information again, we immediately found a 30% increase in the number of identified peptides. And this is due to the fact of this focusing of the ion mobility. Now, the next thing we did was actually not delete the ion mobility information, but selectively extract the information only from the ion mobility window where the peptide is to be expected, and we found another 70% increase in the number of peptide identifications. And finally, when we implemented the multiplexing scheme, we got another 50% increase on top of that due to the increase in sensitivity and the increase in the number of ions reaching the detector. So if we put all of these numbers together, we found a 3-fold improvement compared to running DIA or SWATH on a regular QTOF type of instrument, reaching now about 65,000 precursors. We are now actually pushing this number even further, and we are on the order of 7,000 or more Swiss-prot unique proteins that we can identify at 1% protein FDR, which corresponds to about 92% of the proteins in the library. So this is basically close to very complete coverage of the proteins that we know are present in that particular sample. So with this, I would like to acknowledge all the people who contributed to this work. First and foremost, I'd like to acknowledge the students in my lab, specifically Annie Ha and Max Franco worked on this particular project. And of course, our collaborators in the MAN lab and at Bruker and in the Aebersold lab. And I would also like to acknowledge all the funding I've received to carry out this work from Canada and abroad. And I would like to point out that we are currently recruiting, and we have several open PhD and postdocs positions in my lab. So people who are interested in doing computational proteomics work, feel free to talk to me or send me an email. Thank you very much. Yeah.
Some improvement in identification of peptide. Do you have a way for chip-to-chip accuracy, maybe using some sort of spike samples or spectrum?
Yeah, that's a very good question. So the question is about the quantitative accuracy, and that's definitely one of the next experiments we are planning, right? So we have this very nice setup from the 2016 paper from Pedro Navarro, where we spike multiple organisms in different ratios in a single sample. And this allows us to look at quantitative accuracy on a very global scale. And so far we have not proceeded to that, but that's an obvious next step that we want to do, yeah.
Okay, well then let's thank Hannes one last time, and that'll close the session. Thank you everyone for attending.